All right. So I think for most of us, probably this all started, this whole microbiome, probably started with diarrhea. It certainly did for me. I was using probiotics a little bit 15 to 20 years ago, mainly for diarrhea, using what we had, which were usually human products from the health food store. And so it's amazing what's happened in the last 15 years. So whenever we prepare for these talks, we like to find review articles that might be beneficial to the audience, where other great investigators have pulled together groups of papers and making some sort of definitive statements. One of those groups is Texas A&M with Dr. Sukoldowski, which also works very much with Purina, and he does almost all my microbiome work during our collaborative research. And so I think one of the I like these three statements, so I wanted to attribute them to Jan, is we can now say, I hope you believe, that in small animals there is an immune effect of certain good bacteria that can be enhanced by giving them other good bacteria or other things in food. We now know, and Dr. Quimby is going to be helping us with the kidney part of this tomorrow, is manipulating those bacteria can have effect outside the gut. So when I was thinking only for diarrhea 15 years ago, and now getting to lecture around the world on multiple body parts that microbiome interacts with, it's really kind of cool. We've now seen some evidence for systemic immune. We've seen some evidence today on stress. You'll hear more evidence tomorrow on kidneys. And so it's really great that companies that ultimately will give us products that we can use fund this type of research, because we didn't have it, none of us, right? I'm a practitioner first, a clinical researcher second. So it's very, very cool. So just to remind you of one more term, dysbiosis is basically when that normal plethora of bacteria is no longer normal. That would be a dysbiosis. And so I want to start with the healthy cat discussion. If we think about these great companies around the world that are truly publishing research, doing really good formulation assessments, checking quality control of their nutrients, the big companies, I'll use Purina as an example because they're our sponsor today. If that animal is healthy and they're on a name brand commercial diet that is formulated for growth or maintenance by a good high quality company, their GI microbiome is probably fine. Right? They probably don't need extra supplements if they're normal. And the diarrhea and vomiting, of course, are a measure of normalcy. If they have a nice normal stool and they're not vomiting, I may or may not try to manipulate that animal because they're already healthy. And I'm the same way. Right now, I manipulate my gut if I have an abnormal stool. I'm not going to tell you my Nestle score from today. That's too much information. So remember our definitions. Now we can add dysbiosis to our terms today. So there's been some great work now. In fact, we can even have review articles now that show that those trillions of bacteria, they can be manipulated. And that's the purpose of this nice one from Rachel and Jan, that basically show that if we do some of these things, different nutrient profiles, different protein sources, adding different fibers, adding living organisms that might be a probiotic, we can change that tune, right? And so that's really good. We have to have that background information, right, that shows that it's really happening. Even if you had an animal that you thought needed something that wasn't processed prior to feeding it, which I'm not promoting with this slide, I'm just reminding you that how that group of proteins are processed will also change the gut. Why I care. All of us came, I think, from clinics, or at least most of us, And so before we could measure these trillions of organisms with tools that have improved even in the last year, it's great, because before all we had to measure was, did they stop having diarrhea or did they stop vomiting? And so this now is a new golden age, all these omics and being able to measure things so that we can more scientifically immune modulate with diets. It's just really going to be great for you guys that have 20 to 30 years left in your career. Nutritional immunology, making that microbiome stronger, healthier, directed to help other body parts is really, we're in the infancy, but it's an exciting time. But I don't show heat maps of different bacteria in my lectures. That's about the time I fall asleep in other people's lectures. I don't do it. To me, I like to talk about diarrhea and clinical things. What we might be doing to the microbiome is fine, but I'm not going to have any of those pictures for you today. I want to have a picture that makes the point. Let me ask the audience and Lappin, do I have the labels on the pictures correct? Yeah. So unless you're trying to lose weight, I would say go with the normal stool. It's the better one. And if I see you at the party tonight, if you have a normal stool, come give me a hug. Jason doesn't like hugs, but I'm happy to hug you. If you have a stool that's on the other side today, could you just bow like Japanese? We've seen this in Sandra's lecture already as well, or maybe it may have been Galia's. But point is, for all the slides that I'll show you today, I use the Nestle score. Why I mention 5, 6, and 7 is many of our probiotic studies we're doing with cats in shelters. No one's going to adopt a cat with diarrhea. But I can tell you, the shelter managers, the ones that their job is to adopt cats, not do studies, they are not going to hold a cat in the shelter that has a 4. We classify it as a diarrhea. I would rather personally have a 3, but I'm going to let you know that almost all the stats that I'll show you with studies today, we use 5, 6, or 7. The other reason I do that, especially in the client-owned studies, with clumping litter, most owners can't recognize a 4. You know, they're getting some litter on it, and they just can't accurately score a 4. So I like to use 5, 6, or 7 for those reasons. I really liked these two resources where they summarized most of the probiotic work in gastrointestinal disease in dogs and cats. But is that crazy? How little information we have compared to how many opinions we have on probiotics? You can get all of the papers into 2 review articles. That's crazy. And so that's why when we interact, all the speakers, when we're interacting with great sponsors like Purina, it's just great to get to do more studies. This is the newest one, so I wanted to share this one for you today. If you read in English, they did a very good job with this review. And I wanted to share this one because I also wanted to steal one of their pictures to share with you today. So I think they did a really nice job with their artists. So let's talk about this particular picture for a second. So you see at the top, eubiosis, that would be your microbiome. Gastrointestinal microbiome is fine. Dysbiosis is you have incorrect ratios of many of the different good and bad bacteria. And then if you guys would look at the bottom, biotics, which we've talked a little bit about, the pre, the pro, and the postbiotics, those are going to promote having that eubiosis. In a normal animal, in a sick animal, they're going to drive things back towards normalcy. But one of the things I'm sure you're getting pressure on in your LATAM countries is we probably overuse antibiotics for some things. And we're always trying to find the balance, right, of not hurting pets or people by picking antimicrobial resistance. So we have to be really careful with antibiotics, because antibiotics that you're trying to perhaps treat an ear infection still have to go through the gut to get to the blood. Our microbiome does not like most oral antibiotics, and we can even see that effect with injectable. And so I think all around the world, we're all attempting to use antibiotics correctly, the best way. Like a cat that has ocular and nasal discharge, if it is serous or watery, it's probably herpes. Antibiotics don't treat herpes, right? And so it's doing those things. As we'll learn from Jody, and I can talk about my own work, I just did a 80 cat feline interstitial cystitis study. Of those 80 cats that entered, 1 cat had a bacterial infection out of 80. One study in the United States that I quote all the time, 75% of vets in a big corporate small animal practice gave a shot of a 3rd-generation cephalosporin to every cat peeing blood in the sink, even though 95% of them don't have a bacterial infection. So today's not about having an antimicrobial resistance or appropriate antibiotic lecture. It's about not hurting the microbiome. But that's how we're going to help the microbiome overall of that individual animal, is only use the antibiotic if needed, because we know it can promote dysbiosis. And then, of course, there are pathogens as well, right? Salmonella typhi in cats. Yeah, it's a pathogen, so it's going to make its own dysbiosis. Giardia. Giardia changes the microbiome. Trichomonas, Blackburni, colitis in cattery cats around the world. It induces a dysbiosis. And guess what? Trichomonas, we actually have to give an antibiotic to, ranitazole. And so here we have an organism that's causing a dysbiosis by itself, and we have to give it a drug that changes the microbiome. And so it's really important, I think, for us to talk about these clinical issues and how we can try to lessen that. And then potentially, if we have to use an antibiotic, everyone in this room, I'm sure, has prescribed an antibiotic to a cat in the last 2 weeks, including me. But I'm doing that by picking the best antibiotic and then trying to spare the microbiome, perhaps with some preventative treatments. And that's what this lecture is about today, guys, is taking diarrheas and making them get better faster. And if we have to induce a dysbiosis, say with an antibiotic, is there any evidence that we can lessen that with commercially available products? So, we are getting better tools. I know the same tools are not available in every country. We're focusing on America. Dr. Jan Sukodowski and his team have been able to measure 15 organisms instead of 1 trillion. And they can measure 15 and look at patterns that correlate with normalcy versus abnormal. Whether or not that's needed clinically right now is still debated, and if you're watching Jan, I love the new Dysbiosis Index. But point is, we now have tools for people like Jason, myself, to actually, can we change something with a manipulation like a probiotic? So let me show you a few things to think about. First, before I learned anything about the microbiome, I knew that dietary manipulation made many of my cats get well, right? We don't want to miss parasites, so we do the simple things. And then if they still have diarrhea, or even in acute cases that you would like to get better faster, we frequently manipulate the diet. But now that we have all those studies on diet effect of the microbiome, now I realize when I was doing a diet change, maybe just from one brand to another, maybe from canned to dry, those things were having their positive effect, in part because of manipulating the microbiome. And so it's really cool now to understand why. Remember, I've been doing this for 40 years. I was changing diarrhea with food, not realizing why it was helping. And so today I don't want to lecture about every single pathway for the diets to change. I want to focus a little bit on additives today because we have great probiotic and synbiotic research. But keep it in mind, just doing a diet change can make a diarrhea go away. It doesn't have to be hydrolyzed. It doesn't necessarily have to be a full food trial. Those were great topics from Galia. But in the acute diarrhea cat, sometimes it's just about letting the gut rest for a day and feeding them something different. So bottom line is we know that dietary can work great in the early cases. And then in the more chronic ones, we might continue to do those manipulations with the hydrolyzed novel antigens, et cetera, the elegant work that my friend Galia presented. But I wanted to show you this one real quick. You've probably noticed companies don't often do comparative studies where they try to say ours is better than theirs. They don't do a lot of that. There's not a lot of benefit from that, right? If they're lucky and their diet is better, then they win. But if the diet is equal or their diet is worse, it didn't help them, right? And so this is one I wanted to share with you that's one of the rare studies just to support what I just said. Sometimes a cat that has acute and early chronic diarrhea, just changing diets. These are both excellent diets. They're both excellent diets in that paper, and if you want to pull the paper to learn the diets, please do. But to make the point, going from an excellent small bowel diarrhea diet to a different excellent small bowel diarrhea diet, it was effective. So please don't forget to do the diet change. In fact, a joke, and I think Galia and I say one very similar, is the best diet for a syndrome is the one that they'll eat that works. Not very fancy, right? But that's how I tell. I'm an internal medicine person. It's my day job. I don't know if product X, Y, or Z is going to be the best one for this dog or cat, but changing is not crazy, which is why most universities, we have all three of the big companies' diets, because we do often change. Even in the same prescription class, we'll try a different diet. So keep that in mind. So I've talked about anxiety, stress, and immune modulation. What I'd like to do with the next 16 or 17 minutes is really talk about the GI things. And then tomorrow you'll see Dr. Quimby talk about the kidney findings. So first and foremost, when you're meeting with a company that's trying to sell you a probiotic or a synbiotic, that means it has a live organism. When you're meeting with that company, ask the salesperson to see their quality control data. When that happened in North America, at least 2 different studies now, most of the companies could not tell you their quality. At least the salesperson couldn't. And then when they just bought the products that said they had live organisms, most of the time they did not meet their label claim. So it's a very nice and easy question. I just asked them, you say that this product is good for 1 year or 2 years on the shelf? That's what they say. then say, I would like to see your proof that at the end of the shelf life, there were still living organisms. I'm sorry to use a dog example, but can you imagine how sick these researchers were when they found out in the middle of their study? These studies are hard, right? To find out in the middle of your study that the probiotic group wasn't getting a probiotic. I bet they have gray hair, just like me. So I've seen them advertising the nutrition handbook. I'm so glad that they're doing that. You've seen the QR everywhere. But I wanted to show you my 3 pages in the handbook. I got to write the section on how to pick a supplement. And I really can make it very simple. I want to know their quality control, in particular with the probiotic lecture, numbers of bacteria, appropriately stored, but number of bacteria at the end of the shelf life. If it doesn't meet what we've done all the studies with, then you can't buy that one. These people work hard for their money, and if we're selling them a probiotic that doesn't have a live organism in it, they just got ripped off. And so we don't want that. It's really important. Hold those companies' feet to the fire. They have to prove to you that they have good quality control. I'm going to leave it up to you from number 5 and number 6 if you read in English. You guys get to decide in your own clinics what's the level of evidence. But I can tell you at a vet school that trains 600 veterinarians, the companies, they want to be in our hospital because we're the ones that the students watch. And then the students will say, oh yeah, Dr. Lappin liked blah, blah, blah. That's probably what they're going to buy. So you can imagine all these companies, they want to be in front of the new generation. Well, so my second question for them is, what have you published? You're saying it does X. Why are you saying it does that? And there's great levels of evidence. If 20 of us talk about a probiotic tonight and we agree together that this probiotic that was just released probably works, it probably works. We're a bunch of smart people, right? If 20 of us think it works, it probably does. But the level of evidence for a vet school, to me, they have to publish something. And I really think you guys should push them that way as well, because they're using your clients' money to make a profit. And I think some of those profits should go to prove that their stuff works. And so that fits well with a lecture that I call slide-worthy. In the United States, there are many probiotic manufacturers. These 3 companies have tended to publish the most science, and that's why they get a shout out, because they're publishers. If you have great companies in LATAM that are publishing, that's cool. That's why I gave the others on the lower part. But it's amazing. And if it's not a drug, they may or may not have done a lot of published work. Drugs are obviously much more scrutinized than additives or supplements. So this is a question that comes up still forever. Is 1 better than 5? Is 1 better than 7? Is 1 better than 6 with a yeast? The operative word is better. If somebody says better, what did they have to do? They had to have compared. And this happens a lot with salespeople, and I love salespeople. I want to buy their stuff. But when somebody ever says better to me, I say, what did you do to prove it was better? And most of the time, they're, well, we haven't done, they don't have an answer, right? But you should push them. Don't call it better unless you proved it. Just because there's more, that's an American thing, right? More and bigger and all that stuff is better. Well, that may or may not be true. And so we have plenty of great data from probiotics that have 500 million organisms. And then some of the companies that have 5 or 6 organisms, they have some great data as well. And that's great. But most of the time we can't say better because they haven't been compared. So what I want them to show me as the scientist in GI at CSU is just show me what you've done. And then I'll decide as a scientist, is that enough to get in my pharmacy? So this was my first one with Fortiflora. We had done some of that immune stimulating stuff, the commercial product I just mentioned, but the name Enterococcus lactis, strain SF68. This was my first clinical trial for GI. And so I told the company that makes this one, I said, if you can show me that it lessens diarrhea in the shelter, where there's so much stress and more infections and all that, then we'll consider it for Colorado State Pharmacy. And so the short version, we got one of the really nice local shelters, and we fed the probiotic to one room. The other room got fed the placebo for 1 month. Then we switched the rooms. The next month, the alternate room got the probiotic, and the other room got the placebo. You do that to account for a room effect. Like feral cats, maybe a little bit more stress. In this shelter, the stray cats are next to the barking dogs. And so we switched the month by product to account for a room effect. But if you look at the dark colored bars, those are the diarrhea rates. Cats that had at least 2 days or longer with a soft stool, 5, 6, or a 7. When we added the probiotic, and this was Enterococcus lactis strain SF68, when we added the probiotic, and the far bars are probably the most important, showing combined data, is we were able to drop diarrhea rates from about 25% to around 7% just by feeding a probiotic. And as you know, most shelters can't afford to do a sachet of a probiotic for every cat every day forever. But my point is it proved the hypothesis. A stressed group of animals, and we did do a standardized diet for those 2 months as well, a Purina diet. The point is that if we could prove it in that population of cats, I believe a probiotic can non-specifically help lessen diarrhea personally. This was with another company. I wanted to share this one. This was a nice multi-strain synbiotic, and this is a different type of research. This team at CSU took cats that had fecal scores of greater than 5, and they did an open trial where all the cats got the same probiotic. There was no placebo, so a little bit less rigorous, that type of trial. But there was a 75% client satisfaction. And they dropped from about an average fecal score of 6.6 to down around 4. So just another field study showing that you guys aren't crazy using a probiotic, in this case a synbiotic, in field cases with diarrhea. We know it's manipulating their microbiome, and if that resolves the diarrhea for a fairly insignificant cost, that's a big win for the clients. So I think I picked 3 examples. The cat that is pale has hemoplasmosis and needs doxycycline, the cat with the abscess, and the cat with toxoplasma uveitis. That cat needs clindamycin. We don't have a choice, right? These cats should be treated with an antibiotic. And so I believe in restricting use of antibiotics. But we need to. And so when you need to, can we lessen antibiotic-associated illness, shifting that microbiome to dysbiosis? Can we help that? And you know, there are hundreds, maybe thousands of papers in humans. And I'm going to show you 4 today in veterinary medicine, and that's almost all of them. Yeah, this is still an early place for us to say, could we lessen illness due to an antibiotic? And so we did it with strain SF68. We chose clavulanic acid because I knew from my clinical practice that amoxiclav was about a 35% diarrhea rate, or that's what I felt, was clavulanic acid was about 35%. We did that study, And we decided to give the probiotic first, then the antibiotic a little bit later to let the probiotic hopefully do its work before we killed it. This probiotic is killed by Clavamox. And so you know you have to separate most probiotics from the antibiotic. So we tried to have a design that might work if there's only one human in the family or both humans had to work. And so we gave the probiotic, go take your shower, get ready for work, give the antibiotic later. So that's the design that we chose to use, which is about 60% of the human papers give the probiotic first. And so what we showed, if you look at those top numbers, 23 of 27 cats had diarrhea due to Clavamox. Clavamox is horrible on your microbiome. And if you can do amoxicillin without clavulanic acid, still hurts your microbiome, but not as bad as this one. So take-home message, only use clavulanic acid if you need the potentiation. But then look at the fecal scores. Look at the far side, the smaller. We had no fecal scores of 7 in the group that was pre-treated with the probiotic. And when we looked at the whole protocol, days 1 through 11, the overall stool consistency was better if we gave them the probiotic before their antibiotic. So that paper's been out for a little while, and it was very interesting. If you like actually the microbiome heat maps and all that stuff, we tried to prove why did it work? Why did it work? And that last bullet, mechanism unclear. Well, guess what? The tools have improved. So there was a reason that it worked. We just couldn't prove it with the microbiome tools that we had at the time. So this next information has not been published yet except as an abstract. We took Dr. Sukoldowski's, his Dysbiosis Index, and if you look at the red dots are the probiotic group. Their Dysbiosis Index was improving faster. Day 14 is the most important, was improving faster than the placebo group. So we knew that some changes in those major 15 organisms that are in the Dysbiosis Index, they were actually changing, and it would be because of the probiotic. And these two charts are in the paper that we're submitting this week. So first time I've ever shown this in an audience. We now know that we were improving one of the really important good bacteria because of the lactis, Enterococcus lactis strain SF68. Look at the red dots on day 14. And then, as we all know, primary bile acids are bad, secondary bile acids are good. And so now we have several mechanisms why that probiotic helped manipulate the microbiome and lessened the clinical signs. So good news, the tools are getting better, so we'll be able to publish more science about why probiotics might help. So just to acknowledge another company, this was another multi-strain that was used for clindamycin. And it was interesting because it lessened clindamycin vomiting, but it didn't lessen clindamycin diarrhea. I wanted to just share that with you, is not all pro or synbiotics are going to work the same in the same animal. And so it is okay if you try your favorite one and it didn't help that animal. Trying another good name brand is not crazy, right? Because they do different things. So we wanted to know, because many of my clients aren't buying the probiotic to try to prevent diarrhea. They're not all doing it. I'm recommending it now, but they don't all say yes. And so we wanted to see what would happen if they already had Clavamox diarrhea, or amoxiclav, if they already had diarrhea. And I'll just show you this one chart, and we'll start getting ready for questions. We had no 7s after we started the probiotic, or excuse me, the synbiotic. This is SF68 plus psyllium in this study, a commercial product in some countries. Bottom line is the fecal scores got better faster if we added on the synbiotic than if we used the placebo. And so now I believe that I can probably lessen GI signs due to some antibiotics. I believe that I can potentially treat diarrhea that was induced by some antibiotics. And so are we ready for a meta-analysis with 4 papers? No. But if we keep chipping away with it, we'll hopefully get more data for you over time. Okay, last couple of things. I want you to look at that cut and paste title at the bottom. That's one that Jan and our teams did together, showing that fenbendazole for Giardia does not hurt the microbiome. Metronidazole does. In previous work in dogs, we showed that if you gave SF68 plus metronidazole, they do better than if you give them metronidazole alone. So if you do have to use metronidazole for Giardia, we have shown a positive effect with the probiotic in the dog. I know today's a cat lecture, but at least we have assessed that. If you can use the benzimidazole for Giardia, that's probably a good thing. This is great work from our friends at North Carolina State with Jody Gukin, and it is very clear that ronidazole, which is related to metronidazole, hurts the microbiome. What they were able to show is if this is the tritrichomonas, the microphone, this is the microvillus of the colon. If we can lessen adherence of the pathogens, we believe that then most of their toxins that the pathogens are making get pooped out. Adherence is probably important for Giardia diarrhea and tritrichomonas diarrhea. So I wanted to let you know, if you have a case of tritrichomonas in a cattery cat and you have to use ronidazole, Enterococcus lactis strain SF68 was one of the probiotic organisms used that blocked adhesion, which might be part of the reason that they would get better faster. So now when I use ronidazole, I also prescribe that probiotic. I said this in the introductory remarks, so we won't belabor it, but when I was you, young people, I was sending home parvo animals on antibiotics. That was totally wrong. Once their fever breaks, once they have a normal stool, they don't need an antibiotic, they need a probiotic. So please discharge parvo, dog or cat, with a probiotic. And then lastly for this one is we have started looking at manipulating the microbiome by to treat or help cats with constipation. Slab 51, which is in a commercial product around the world, actually has a product now in some countries showing a positive effect. Our work is with SS68 with psyllium, and that study is ongoing, so I can't give you final results yet. But I'd like to show Mia, because she was our index case, that when we fed her the probiotic with psyllium, she actually had her fecal score soften and her creatinine dropped. And so that is an ongoing study that we based on this cat. And if I ever get invited back by Purina, I'll share the data when it's published. But I do think it's something to consider, right? We know that the microbiome affects constipation as well. And obviously psyllium can also do a little bit of stool softening. So I hope we covered all the topics that I wanted in that 50 minutes, and I think we're ready for questions. You'll have to get a microbiome transplant lecture in the future. Thank you very much.